Hello everyone. In this lecture we will discuss about the particle size distribution. So the percentage of various sizes of particles in a given dry soil sample is found by a particle size analysis or mechanical analysis. By mechanical analysis is meant the separation of a soil into its different size fractions. So the mechanical analysis is actually performed in two stages. So first one is sieve analysis and the second stage is sedimentation analysis or wet mechanical analysis. And the first stage is basically meant for coarse grained soils only, while the second stage is performed for the fine grained soils. So you have to remember this one. So sieve analysis we prefer for basically for coarse grain, and the second sedimentation analysis we prefer for fine grained soils. But in general, a soil sample may contain both coarse grained particles as well as fine particles, and hence both the stages of the mechanical analysis may be necessary. The sieve analysis is, however, the true representative of grain size distribution, since the test is not affected by the temperature. So first is the sieve analysis. The complete sieve analysis is again divided into two parts: fine analysis and coarse analysis. And in the Indian standard IS 460-1962, the sieves are designed by the size of the aperture in mm or in millimeter. So the sieve analysis is termed for the particles having size greater than 75 micron or 0.75 micron. In that case, we prefer sieve analysis method to segregate or to distribute the different particle sizes of soil. On oven drying, the sample is separated into two factors by sieving it in 4.75 mm IS sieve. So the soil portion retained on 4.75 mm IS sieve is called gravel fraction and is kept for coarse analysis basically, and the soil portion which are passing through 4.75 mm IS sieve is subjected to fine analysis, or these are known as fine grained soil which are passing through this 4.75 mm IS sieve and which are retained on this sieve 4.75 mm IS sieve. These are known as coarse soil or coarse grained soil or gravel fraction. Next, the set of sieve which we use for the fine analysis are Indian standard or IS 2 mm, 1 mm, 600 micron, 425 micron, 300 micron, 212 micron, 150 micron and 75 micron. So these sets of sieves basically we use for the fine analysis according to Indian Standard Code. And another one, the set of sieve which we use for coarse analysis are IS 100 mm, 63 mm, 20 mm, 10 mm and 4.75 mm. So this set of sieves we use for the coarse analysis. Next is sedimentation analysis or wet mechanical analysis. The sedimentation analysis or the wet mechanical analysis is conducted on soil fraction finer than 75 micron, which is kept in suspension in a liquid, that is usually water medium. And the analysis is based on Stokes' law, according to which the velocity at which grains settle out of suspension, all other factors being equal, and is dependent upon the shape, weight and size of the grain of the particle. However, in the usual analysis, it is assumed that the soil particles are spherical in shape and have the same specific gravity. So with this assumption, the coarser particles settle more quickly than the finer ones. And the sedimentation analysis is done either with the help of by using a hydrometer method and a pipette method. So these two methods are used to find out the particle size distribution in sedimentation analysis. And in both the methods, a suitable amount of oven-dried soil sample finer than 75 micron size is mixed with a given volume V of distilled water. So in this case or in both the methods, we have to add the soil, oven-dried soil, particularly the oven-dried soil which are passing through or finer than 75 micron is mixed with a volume of water. This water is distilled water we have to take this procedure. The mixture is shaken thoroughly and the test is started by keeping the jar containing soil water mixture. And we have to keep it vertical. This soil sample, we have to keep it vertical. And at the commencement of sedimentation test, soil particles are assumed to be uniformly distributed throughout the suspension. And for this sedimentation analysis, there is some limitation which we have to follow during the continuing this procedure. So limitation are, as the specific gravity of solids for different particles is different, the use of an average value, we have to use an average value of G in a source of error. However, as the variation of the values of G is small, the error is negligible. So we have to take an average value of G. Next, the sedimentation analysis cannot be used for particles larger than 0.2 mm as turbulent conditions develop and Stokes law is not applicable. So we have to take care of it that the soil particles may not be larger than or greater than 0.2 mm. Next, the sedimentation method is not applicable for particles smaller than 0.2 micron because Brownian movement takes place and particles do not as per Stokes law. So again, if the particles are very small like 0.2 micron, then also we cannot use these particles to take this procedure. Also, the soil particles which you take must be in spherical in shape and the particles settle independent of other particles and the neighboring particles do not have any effect on its velocity of settlement. So we have to take care of it. The settlement of the particles is influenced by the surrounding particles as the liquid is not of infinite extent. The particles falling near the wall of the jar are also affected. So during this procedure, we have to take care of this. So let us discuss the first method, pipette method. The pipette method is the standard sedimentation method used in the laboratory. So it is a laboratory method. The equipment consists of a pipette, a jar, and a number of sampling bottles. Generally, a boiling tube of 500 ml capacity is used in place of a jar. So we use a 500 ml capacity boiling tube in place of jar. So the figure shows a total arrangement. This is the sampling pipette within which we keep the distilled water with soils or mix, and here we keep within this instrument, we keep this instrument vertical or we keep this pipette vertical. Already I have told about it. So this is the total arrangement of this instrument. The figure shows a pipette for extracting samples from a jar from a desired depth, that is HE. The pipette consists of a 125 ml bulb with stop cock for keeping distilled water and a 3-way stop cock suction and wastewater outlets. Sampling pipette of 10 ml capacity are used here. The method consists in drawing of samples of soil suspension 10 ml in volume by means of this pipette from a depth of 10 centimeter at various time intervals after the commencement of the sedimentation. So again here we have to take care of this. So basically, to prepare the method of soil suspension in the sedimentation analysis, only those particles which are finer than 70 micron size are included. Hence, the soil sample is washed through a 70 micron sieve. About 12 to 30 gram of oven dried soil sample is accurately weighed and mixed with distilled water in a dish or beaker to form a smooth thin paste, and we have to keep this paste within this pipette. So the recommended time intervals are half, 1 minute, 2, 4, 8, 15 and 30 minutes, and 1, 2, 4, 8, 16 and 24 hours record from the commencement to the test. The pipette should be inserted in the boiling tube about 25 seconds before the selected time interval, and the time taken for sucking the sample should not be more than 10 to 20 seconds. And each sample so taken is transferred into suitable sampling bottles and dried in an oven. And the mass, after oven drying, the dry mass MD of solids per ml of suspension is thus found by taking the dry mass and dividing it by 10. We have to divide it. So this much, and in next lecture we will discuss about the next method. This is hydrometer method. So thank you.